Good afternoon, everyone. Uh, well, as he mentioned, my name is Gastón, and I'm going to talk about our research entitled Reproductive Disorders and Contaminant Exposure, and how it relates with the variability in isotopic signatures in the amphipod Monoporeia affinis. So, why we wonder this? About the physiological state effect on isotopic variation on these animals. As you may know, stable isotopes analysis is a widely used tool for ecological and ecotoxicological research. For instance, by studying the dietary niche of an animal or the bioaccumulation and biomagnification in contaminated areas. And this approach states that an animal, a consumer like this one, the signature of the stable isotopes stems from three main components. First one, the food sources. It means what they are eating. So they are receiving information from that. Then how they get that food. Yeah. And the third one, it's how it's incorporated in the tissues after the physiological processes, different physiological processes, right? But surprisingly, this last one is usually less considered less influencing in the whole signature of the animal. So this came to the main core assumptions of the use of stable isotopes that states that non-dietary components are negligible compared to the food sources. And in the end, isotopic niche are reflecting just the food sources and the foraging behavior of a consumer. But there is a lot of research, experimental and modeling research, that indicate the opposite, that physiology actually has a main impact on the stable isotope variation. For instance, this variation was related with food deprivation, parasitic infestations, and also exposure to contaminants, to chemical contaminants. So we wanted to link this physiological stress or physiological state of the animal with the variability in the stable isotopes. So we focus on the amphipod Monoporeia affinis in females that were collected in two different areas of the Baltic Sea coastal area. One was Askö and the other one was Bråviken. That were considered one as an uncontaminated site due to the low contaminant levels after the effect screening survey. And the Bråviken was considered contaminated due to the high levels of hydrophobic organic contaminants as PCBs, PFAS, and heavy metals. And we focus on Monoporeia affinis because it's a species used as a biomonitoring species in the Baltic Sea. Because it is well established the relationship between environmental stressors and biological responses, especially reproductive disorders. Yeah, and actually these responses are usually well correlated with different kind of contaminants. So we established the health status of females based on the amount of on the percentage of embryo aberrations that were carrying the females, and we classify them as healthy if they has less than 30% of malformed embryos, and unhealthy if they were carrying more than 8% of malformed embryos. And all these females from both sides, we measure the composition of carbon and nitrogen, and we use this data to estimate the isotopic niche metrics proposed by Layman. And by doing this, we expected to see higher variation in the isotopes in unhealthy females, especially from Bråviken, that was the contaminated site. So what we found? First, looking at the single variation per each stable isotope, we see depletion on carbon in unhealthy females from both sides, and also an enhancement and an enrichment in nitrogen also in unhealthy females from both sides. And looking at the joint variation of both isotopes, we see these slopes are steeper for unhealthy females, especially in Bråviken. And then we suggest that this is due to oxidative processes, probably linked to contaminant exposure. And actually, this was supported by biomarkers related with oxidative stress, like lipid peroxidation and oxidative reactive compounds. And so it is actually showing us a response on a stress condition. So now, looking at the niche metrics, we wanted to see if there was an effect on site. So first we have the healthy females from Askö, and then we have the healthy females from Bråviken. And we can see that their niche are overlapping, and we only see a small variation in the orientation of the ellipse. But if we look at the standard ellipse analysis, we don't see any difference between both groups. And now if we look at the unhealthy group from the reference site and the unhealthy group from the contaminated site, we see in both cases that an apparent extension of the isotopic niche. And actually there are not difference between them, but there are difference between the healthy groups. And if we consider the traditional way to understand isotopes without taking care of the physiological state, we can conclude that these animals enlarge their isotopic niche. They are having like more food resources because we see an expansion in the carbon axis that would be interpreted like more food sources. And also there is changes in the trophic position because we have an enlargement in the nitrogen variation. But if we take a look to this part, here we have the isotopic baseline data for the sediments that actually are the food sources for these animals. And we don't see any differences. So we cannot interpret this like they are changing their food sources. Neither the behavior in feeding, because actually it's the same species, and they feed in the same way. So what we did was to combine these groups, just considering the health status, and of course we observed the same pattern. Unhealthy groups show a wider niche, that although they overlap, the standard analysis show that they are significantly different, and yeah, we can actually attribute this difference to the health status. So, what we learned from this experience, actually, isotopic variation was related to the reproductive disorders that we use as a health proxy for the physiological state. And we observed that isotopic variation was higher in unhealthy females that were carrying embryo aberrant. Also that the isotopic niche changed. They became wider. And in both cases, they were characterized by depleted values of carbon and higher variation in nitrogen. So isotopic signatures and isotopic variability can be used for addressing the stress conditions of animals. So as a take-home message, we conclude that non-dietary component actually has effect on the stable isotopes variation and should be considered in food web studies and trophic position assessment. And also that can be used to infer the health status of Monoporeia affinis together with biomarkers and this embryo aberration analysis. Okay.
Thank you.